Happy Friday, everybody. We're doing it COVID style. So we're switching things up a little bit. It's me, Teresa.
And me, Mandy.
We're here for Food Safety Info Friday. So we're going to be mixing it up a little bit. And so here at River Ridge, we have been blessed by the beautiful Mandy Leitz. And we are so thankful that did I say that? I said right? So we are so thankful that she has joined the River Ridge team. And so Mandy is a lab technician here at River Ridge. And so we're going to kind of just get a little background on Mandy. So could you tell us a little bit about yourself, Mandy?
Yeah. So I grew up on the east side of the state. So I'm from Clinton Township, which is about 20 minutes north of Detroit. And then I went to college at Ferris State up in Big Rapids, where I got my Bachelor of Science in Biotechnology.
Go Bulldogs!
Go Dogs!
How awesome! So what is it here or what do you do here at River Ridge?
So as we all know, the lab is located up at Cider, and so I work with quality control up at Cider and making sure the cider tastes good, that it's safe for the customer to drink. And then I also work with River Ridge Farms to help them design their trials for the growing season. And then I work with the packing sheds on their environmental monitoring program.
Since you brought up environmental monitoring, so that, as you know, is a big thing in the pack houses or the processing plants, that everyone should have an environmental program, and we would like you to have a strong one, right? So the biggest concern, it's a concern at processing, but since they have a kill step, it's a little bit different than the fresh side of things. So listeria is a big thing. So could you kind of give us an overview of what listeria is? Because some people might not even know what that is.
Yeah, let me do this real quick. So that's true. I feel like we hear about listeria a ton, but do we really know what it is? So hopefully this will give you a little bit of answers as to what is actually going on with listeria. So we're gonna start out basics. So Listeria monocytogenes is a subspecies of the genus Listeria, and there's 15 species in the Listeria genus, but only monocytogenes is the harmful one, and that's the one we're concerned with. It's a facultative anaerobe, so fancy word for saying that Listeria can live without oxygen. It's a gram-positive bacteria. Now you may be saying, what the heck is that? And why does it matter to me? Well, gram-positive bacteria have a really thick cell wall made of peptidoglycan, which is sugars and amino acids. So this kind of relates to your chemical concentrations and making sure that they're at the appropriate concentration so that they work effectively to either destroy the cell wall or get through it. So down here we have a basic structure of a Listeria cell. So in here we have the DNA, which is the basis for all living organisms. It is what makes you, and you, me, and makes us function. This is like a tail that the Listeria has. It's called a flagellum, and it basically helps the bacteria move around in water and on surfaces. So the next question is, well, how does it reproduce? How does it grow? So Listeria is a non-spore forming bacteria. And what that means is it replicates by simple binary fission, which means that you have your DNA in the cell, and then it replicates into two sets of DNA. So you take one DNA, now you have two. Then your cell wall and your plasma membrane will split down the middle. And it will keep splitting until you have two separate cells, each with its piece of DNA. And over here, we can see an actual microscopic picture of a Listeria cell that is actively dividing. We can see the cell wall here, kind of pinching off the two sets of DNA. All right, so the next thing is where Listeria live. Well, we know it likes cool and wet environments, so it gets introduced into our facilities through either soil or employees. It can be in rotting fruit, and it can be in healthy fruit too. But maybe we often don't think of Listeria being on good-looking apples because, like, oh, they look fine, but they actually can have Listeria on them. So as we said, found in soil, prefers wet environments over dry environments. That's why they're more of a problem in your wet end than in your dry end. So they also can be incorporated into biofilms. So what is a biofilm? It's basically a film of bacteria that they all kind of get together and say, hey guys, let's protect ourselves from these people that are trying to kill us. So you can see over here, this is a microscopic image of a biofilm that formed on this meat cutter. And you can see there's a space, here's a stainless steel. So we can see, it's a pretty hefty film it's forming. So Listeria monocytogenes in particular can grow between 86 and 98.6 degrees Fahrenheit. And that's its optimum growth temperature. So those are the temperatures that it really likes to grow. But it can grow in temperatures between 33 and 113 degrees Fahrenheit. It won't grow as fast as if it's in the 86 to 98 degree range, but it'll still grow. And then it can survive freezing, refrigeration, heating, drying. Listeria is pretty tough. It's a pretty tough little guy. It can also grow and live in pH ranges of 4.1 to 9.6, and it can even survive outside these ranges. And then this is a Listeria species culture. So each of these dots is a single Listeria colony. And it can live in pack houses for over 10 years if not taken care of properly.
That is so interesting. I didn't know that it could live for 10 years in an environment. That is simply a very hard guide to get it once you get it into the building, right? Mm-hmm. So since we know that Listeria could be in our pack houses, on our line, anywhere, we implemented in our environmentals to swab. So we get swabbed by a lab, and then a couple years ago, we introduced a kind of rapid testing at River Ridge that we can do. So we had a Certus, and now this past summer, we got the Listeria right now from Neogen. So this gives us test results right away, and you're kind of the one that has been doing all the testing with this and getting all the results in the lab for us. Could you give us kind of a little bit of an overview on this Listeria right now?
Yeah, so Listeria right now, first it can give you results in 20 minutes after you get the sample all prepared. So it's a PCR-based method, which stands for polymerase chain reaction. And then it is solely based on the emittance of light from the sample. So this determines whether you get a positive, a negative, or an invalid. So, but we will dive into this topic on how it works exactly in one of our upcoming sessions.
That is so cool. So you stated that it can give you a positive, negative, or invalid. So positive, negative, we know what those are and kind of know what to do with them. But an invalid, what would you suggest that we do with an invalid, and why does this machine get invalids?
So yeah, that's a good question, because I feel like this season this is first season we've really started to use this machine, and we didn't really know what do we do with this. So an invalid simply means that the machine could not read the sample. And there are a couple reasons why the machine can't read the sample. The first one is dirt. So these are all samples that were run in the lab here, and they are samples from various pack houses that River Ridge works with. So in each of these circles, we see that, there's a pretty good amount of dirt. And in each of these tubes, there is only 500 microliters worth of volume. So, I mean, this is a pretty substantial amount of dirt for that tiny amount of liquid. That's half a milliliter. Another reason why is wax. So in this blue circle, we can see that this was a wax sample taken from a wax room. And part of our process is heating up the sample. So when it does that, it kind of forms this pasty white mixture that is like chunky. It's not liquidy. It's almost like a solid. So that will prevent light from getting through to for the machine to measure. A final reason why a sample could be invalid is sanitizer. So some obvious samples that have a sanitizer issue are ones that have bubbles all over the place. So if a sample has a ton of bubbles in it, the machine can't read it properly. Another sanitizer sample could look like these samples. These are perfectly normal samples. None of them came back invalid. But if there's a lot of sanitizer present but doesn't form a bubble, it can overwhelm the machine where it can't read it. So what do you do if you get an invalid? Well, in the case of dirt and wax, if it's too dirty to read, the surface probably needs to be clean. So that first of all, we can get an accurate reading, and so that you're keeping up with your sanitation program. With sanitizer, maybe go check out the area and see, was there excess amount of sanitizer? Did we just clean there and didn't rinse it all away? You know, kind of visualize it out, and if it needs to be recleaned, then reclean it.
And sometimes you think that maybe they are overusing sanitizer and they don't need that much.
Yep, so like in our last video, we talked about Sterilex powder, and I was doing the correct way where you just lightly sprinkle it, but Teresa was doing it where she was just dumping piles, and you don't need that much sanitizer. If you do it correctly, it will do the job. If you dump it, you're just wasting, first off, chemicals, and you're pouring money down the drain because you don't need that much to do the job.
So, Mandy, you have been going into these packing facilities at least once a month doing these testings for Listeria right now. You've been in on the Cornell project and been swabbing underneath these equipments. And you've got a good look at all of these pack houses. What is some advice that you would give these packers for their environmental programs?
A seminar a couple weeks ago, and the speaker made this comment. He said, a negative result does not indicate food safety. It means that the food is safe for that test. I think this quote is important to keep in mind when we think about our environmental monitoring programs, because when we do a test, we do it at a specific time, on a specific day, at a specific spot in our huge facility. And, you know, and that one test does not represent your entire facility. So that's important to keep in mind. You know, we test a super small area, and for our monitoring program, only 5 spots, to kind of give you a representation of your whole facility. So it's important that you guys make a list of different spots each month, and that you rotate through them spots each month, so that we can get a bigger picture of what's going on in your facility and kind of tell your story of, you know, what's going on. But when you do get a positive, don't be afraid. I know it's scary. Don't be afraid. Just go at it. Remember to go out with a calm approach and just clean and scrubby. It will be okay. We promise. Because me, Teresa, Jamie, we're all here to help you get through it.
That is so true. That is so true. Another thing I want to ask you is, what is advice for these packers or processing plants for the sanitation?
Yeah, so just over the past couple months, what I've observed, there's a couple things. So I'll start out with an example. So we all know Michael Jordan, greatest basketball player of all time, arguably. He did not spend half an hour a day in the gym shooting free throws, 3-pointers, whatever. No, he spent like hours a day, and he worked his butt off to get where where he's at today, to be called one of the greatest of all time. Same thing kind of goes to your pack houses. Yes, it's not basketball, but and it's not like you're in a gym shooting free throws, but it's how you clean, how you take it. Do you want to take it seriously? Are you going to take it lightly? And you can get your facility to be one of the best pack houses in the state, or who knows, in the country. Anyways, so one thing I think we could focus on is time. How much time you spend cleaning. You don't want to spend an excessive amount of time cleaning, but you also don't want to spend too little. So it's kind of finding that happy medium. So an example is taking a 4-minute shower with no scrubbing versus an 8-minute shower with soap and scrubbing. So you jump in Lake Michigan, we all know it's kind of nasty in there, a lot of fish swimming around and who knows what else. You get home, hop in the shower, and you just rinse off. Okay. Then you go take an 8-minute shower with soap and water and you're scrubbing. Which shower are you going to feel cleaner afterwards? The shower where you rinsed off, or the shower that you scrubbed yourself with soap, make sure you were all clean? I know for me, it's going to be the 8-minute shower. Same thing goes with your equipment. Give it that time that it needs to get fully clean. So with that comes scrubbing. So scrubby, 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 always. An example is washing dishes. You wash your dishes at home and you scrub them to get the food off. So why don't you scrub your equipment that has rotting apples on it, or your leaves, or what have you on it? So make sure you scrub. Scrubbing is very, very important. Next, chemical concentration. Make sure you are using your chemicals to what they are supposed to be. So an example is the sugar in cookies. So we have a recipe. It calls for 1 cup of sugar. When you make the recipe, you put 1 cup of sugar in it because that's what's going to make it taste good. You don't put half a cup or 5 cups. It's not going to taste right. Same thing with the chemical concentration. Like I said in the beginning, the Listeria has a really thick cell wall of that peptidoglycan, and we need that concentration to be what it's supposed to be at, so that it can either penetrate or destroy the Listeria altogether. Finally, you can do training. So maybe this is integrating a weekly training where 1 week you focus on cleaning the brush beds, making sure people are trained on and they know how to do that. Then the next week you work on the wax area. Then the next week you work on floors. It's kind of giving that people a constant reminder of how to clean, make sure they're doing it right, and always living up, always doing what they're supposed to be to the high standard that you want. And then you could integrate monthly meetings, monthly food safety meetings, where you talk about Listeria results, problems, environmental problems, and just emphasizing the point of why food safety and cleaning are important.
Yeah, I like that. And they could even not even call them food safety meetings. They could just be meetings, and they could bring production into it. They could bring in shipping into it. They could bring in maintenance into it. So make it feel like it's just not a trigger on the food safety, right?
Make it whatever you want, as long as it gets the job done. Have that open communication with everyone, so everyone kind of has somewhat of an idea of what's going on. Bring donuts.
Yeah. Oh, I like donuts. Well, thank you so much, Mandy. It was so very much informational, and I'm glad we got to talk about this COVID style. And so we are actually going to be doing this every other week. So we're going to have Food Safety Info Friday. And the next one, we're probably going to be talking about Listeria right now compared to a Summit Laboratory or a certified place that does swabbing. So it's the PCR versus a cultural Listeria test. It's like comparing apples to oranges, like Mandy would say. And we're going to have a little bit of biology 101. And then we are probably going to do ATP testing and ATP trending, which we do want to dive in deep with trending too with you guys. So thank you so much for joining us, and we hope you guys have a great Friday.
Bye.